let's look at, and we can sort this by any way. So we can go to, say, day, p-value, and cell type. You can right-click on that and go sort ascending. Does everybody did Meg 3 come up for everybody? Yeah. What is Meg 3? What does that stand for? If you click on gene assignment, you'll see the name of it. Does everybody see a name on that? Maternally expressed 3. Remember, these are from different cell types. What's happening is there's probably a gene that gets expressed in certain... So here's what happens with DNA is basically when your mom, gives you your DNA, your dad, a lot of times whatever they have compressed will stay compressed. This is one of these genes where it has that variable position. So what's probably happening is this is an area where in some individuals it's more compressed. That means you don't get as much gene expression, and in some areas it's more wide open. Which makes sense for this kind of cell type. I think this is awesome. You can click on this, so we can select. So select row 1. Right-click and go dot plot. So this would be the relative expression for MEG3 divided by day. Times 0, and then we can actually, what we can do, go to shape. So up here in the menu where the colors are, instead of saying color, go shape. And we could say, I want the cell type color. So we can see this gene. We can actually kind of confirm that we're seeing differential expression. That not only is it affected by the different cell types, but it's also affected by differentiation state. You can see there's very different levels of this gene at baseline before these cells are induced. Once they're getting induced, these stay the same. These actually go down. And again, this is just one gene. But a lot of times I'll be on a disease, and you can actually just go down the list, like H19 imprinted maternally. Something's going on. One of these cell lines got, some condensed DNA from its mother. The other one did not. And we're seeing this in the different cell types. Will this have any effect on differentiation states? Probably not. But I guarantee you there's a lot more in here that do. Yeah. Gene names. 1, 2, 3, 4. RefSeq? Oh. So when we attach the annotation file. So if you look at your original column, here are gene names. Wait a minute. All our basically Affymetrix, instead of putting gene names, what it does is put a number because it makes life easier for them. Because they can put a number for the same gene, a different number for the same gene on different arrays and keep them separate. So what Affymetrix does is just assigns a number to the genes. Before, what we did was we attached a file to say anytime you see this number, it means this gene. So as soon as we did the ANOVA and annotated, it basically took that number and goes, oh, that's MEG3. And it gave us that. Does that make sense? This is all from the original file. Okay, so we showed how to display this, basically a dot plot. Now that we got the numbers, we can take a look at all kinds of stuff. How do you think when we looked at the PCA, we pretty much knew that we were going to find lots of differences, right? There was no doubt about that. When you summarize the data and they basically end up in three, distinct areas in three-dimensional space, something's different. Here is another way you can tell if basically your experiment worked. Highlight the p-value day times cell type. That'll be column 8. I'm going to right-click, and I'm going to go plot. Select histogram. Now hit OK. Lovely beta curve. So what this is a distribution of the p-values. What you can do is you can make these bars a little more narrow. So if you saw what I did, hit that red plot properties again. Here under histogram options, instead of bins 20, make that 100. You can make it 200, anything you want. Hit apply. What this is telling me is that I have a high proportion of low p-values with this test, right? And this goes down as we get higher. In statistical terms, this is called the beta curve. If you plot your distributions of p-values and you notice that you have this curve, that you have a higher proportion of p-values at low p-values compared to everything else, things changed. If I did this for the day 0, day 5 comparison, this p-value would be even steeper, or this beta curve, right? Kind of a quick and dirty way to basically see, whether it worked. So we can see if it worked. What we can also do, another way to do that is go to workflows. Select the first one, gene expression. I love this. This is a cool function here. Go to analysis. Oh, let me see it. There we go. So make sure you're not selecting a column before you do this. Hit plot sources of variation. Anybody know what an F ratio is? I'm going to punch Vitaly in the face. He didn't teach you anything. Actually, I don't think I will. He'll probably kill me. He's got all that macabre gras stuff. Well, don't feel bad. I had to look up F ratio too. Like I said, don't sweat the details, guys. What an F ratio is basically, it's kind of taking it's the variance compared just to the random variance. So it's the variance due to your model versus the entire variance in the system. So that basically you can consider this F ratio the times more above random noise that your variable or your characteristic or your model is introducing into the system. As you can see, so the error rate is always 1, right? Anything above 1 it will be a significant effect. As you can see, day and cell type, I know it's hard to read, that's about 2. So it's about 2 times above random noise. So we are seeing significant changes due to both cell type and due to the differentiation state. If you just look at cell type, that's about 4 above. Now this, 34 times. This is your mama, this is the variable that's really contributing to your data set right here is the red. And you can see why that might happen, right? You're taking an undifferentiated cell and then you're turning it into, say, skin or endoderm, I think in this case. You've got a cell type with not very many genes turned on, and then you turn on a bunch of different genes. That's a huge, huge difference, and it really shows up in your expression data. As far as me getting any kind of meaningful thing out of the day, that would be difficult because it seems like almost everything is changing. But here, cell type, and then this cell type and cell type and the day, I think we can really work with. But I think this gives you a good chart, and you could do pie chart, sum of squares. Again, showing day is, the overwhelming kind of variable. And you can, you can print all this, yeah. Because with the cell type, they said that, and you saw at day 0, those cell types they're kind of in the same kind of three-dimensional space. So they're not really different. The biggest variable in that whole thing was, when you look at that graph, like you have day 0 here and then day 5 way over here. It's a difference between maybe, a pluripotent and embryonic, which are kind of similar, versus, an embryonic cell versus like an endoderm cell. And sometimes like this, it depending on what you're looking like, especially with like, I do a lot of disease stuff, and I always hate doing anything having to do with the inflammatory response. And it never fails. Like, leave it to researchers to go way overboard on the stimulus, right? Instead of just poking my system, they hit it with a sledgehammer and they go, Mike, tell us what's going on. I'm like, I don't know. You broke everything, man. You hit it too hard. If everything changes, then it's really difficult to determine, what you should really concentrate on. But I think what you're going to find is you can subdivide it and kind of create subcategories that I think are very informative. All right.